All right, good morning. So now we're going to be looking at, or good afternoon, we're going to be looking at chapter 8 here. We're going to be looking at microbial genetics. All right, so genetics is the study of inheritance, the heredity of living things, how traits are passed on. We're going to look at things, or this covers things like transmission of biological property, which is those traits from the parent to an offspring, how those traits are expressed in an organism, which is the phenotype, the structure and function of the genetic material, how genetic material can change, such as a mutation, how we manipulate and change genetic material through genetic engineering. And so that's the field that's got different techniques by which we are able to manipulate DNA in the lab. So it's really pretty cool, has some pretty cool applications. All right, so after we go through transcription, we then have to go through translation, and we're going to move from the nucleus into the cytoplasm of the cell because that's where this ribosomal subunit is located. So ribosomal RNA in bacteria is a little smaller than eukaryotes. It is 70S. In bacteria, eukaryotes about 80S, which we have different sizes, okay? So each ribosome is going to have this large subunit, as I've already said, and then a small subunit. That's the blue and gray areas. Okay, those are made of RNA and protein. Bacteria can have up to 20,000 of these all actively working at the same time. All right. Who are the key players in translation? So during translation, everything that is needed to make or synthesize a protein from RNA, from mRNA to the amino acids, so that's including all of those things, are going to be brought together here at this ribosomal subunit. So this is a key place here. This is where the action is going to happen. So you're going to assemble the small and the large subunits in a very specific manner, and it's going to create very specific sites that holds the mRNA and the two tRNAs with their amino acids. And so we have 3 sites on there here, okay? So this is the small subunit. This is representing the mRNA. It's going to feed through here almost like a ticker tape, or if you have one of those label makers where the paper just shoots out, it's basically like that. And then you're going to have the E, P, and A site, and you're going this direction. So E is actually going to be the exit site. So, and we're going to get into some details. I'm getting a little ahead, but your tRNA is going to read the code on the mRNA. It's going to have a matching 3-letter codon that's going to say, hey, go and pick up this particular amino acid. It's going to pick it up and bring it over here. This little purple thing is representing the it's actually going to bring it here. The little purple thing is going to represent the amino acid. It's going to bind here. It's going to slide over. It's going to leave the amino acid. It's going to exit, and it's going to leave. Okay, and so we're going to get into those details. All right, so I've talked a little bit about these 3-letter codes, these codons. These are very important, okay? These are going to be what we code from the language of DNA. We go to the language of RNA. And so these are in the language of RNA, and they are going to basically code to the language of amino acid. And these are universal with like one exception, which would be methionine, which is going to be formal methionine in bacteria across all organisms everywhere. All organisms, which is really pretty fascinating and actually serves to support the idea that we all come from a universal common ancestor. Okay, so a codon is this 3-letter triplet code here. Okay, it's 3 little nucleotides, UUU, use uracil, right, or UUC or UUA, and that 3-letter code is going to tell you which amino acid to get, or tell the transfer RNA which amino acid to get. What is the order of amino acids to make this specific protein, okay? We have 64 different triplet codes that we can come up with these 3 little letters, but we only have 20 amino acids, okay? That's important. Some amino acids are represented by several codons, and we call that redundancy. And that is important because it actually gives us a lot of protection when it comes to mistakes being made in DNA. So if I accidentally copy, let's see what color is going to work well. Let's say my code says, have you ever made a typo when you type? I'm going to go ahead and age myself a little bit here. When I started typing, we didn't really have, they had microcomputers, but most people didn't have them. And so we had typewriters and we had this little tape we had to go back and fix. And if you made a mistake, it was so hard at the very beginning. You had to like retype everything. Okay, so you did not want to make mistakes. So if I accidentally typed UAA, okay, you can see here UAA codes for leucine. That's an amino acid. So this is the codon, all right, and this is the amino acid, okay. Now it's also showing you the 3 positions, so the order that those go in. So I can code for leucine in I can code for leucine as UUA, UUG, CUU, CUC, CUA, or CUG. There are 6 different ways that I can code for leucine. So I can kind of make some mistakes and still get leucine. That is redundancy. Okay, so if I accidentally type UUG instead of UUA, I'm not going to get a mutation. All right, I'm still going to get the correct amino acids. Okay, now we have what we call wobble, and that's when only the first 2 nucleotides here. There's 3, there's 2 here. The first 2 nucleotides are required to encode the correct amino acids, and the 3rd nucleotide does not change its sense. In other words, it's not a mutation, which is the example I just gave you. That is what we call the wobble effect. And so that allows us a little bit of variation or mutation without changing the message, which is very helpful. So if a DNA sequence is known, your mRNA codon can be figured out from that. You can kind of work this both ways. It can go, and this is how retroviruses work. You can go either direction, right? If it's a code, you can go in the forward direction or the backward direction, okay? So if a codon is known, the anticodon, which is the matching opposite, okay, the anticodon, and so tRNA here has the anticodon. So if you have AUG, you match these up. A is going to match with U is going to match with A, G is going to be with C. Well, if I have UAC, I can, of course, match that back the other way. I can say, well, that's going to be A is going to be U, C is going to be G, okay? So the reverse isn't quite as straightforward, but it works, okay, because of redundancy. So when you have a redundant code, you may not be able to get exactly the same if you are down here at the protein amino acid level. All right, so if you were at the amino acid sequence level, due to the redundancy of the code, it's a little harder to work further backward, okay? So, NCLEX prep question. So the function of a codon in protein synthesis is best described as a 3 nucleotide sequence, which does what? Specifies the initiation of translation, specifies the termination of translation, is on tRNA and is complementary to the mRNA code, or it dictates a specific amino acid. And let me tell you, in this class or any science class, vocabulary is always vitally important. So pause this and come up with your answer. So a codon is going to be D. It is going to dictate a specific amino acid, okay? So here's some more of these special adaptations. I say adaptations, but the more efficient ways that bacteria have in order to work a little faster than we do. So in bacteria, mRNA translation starts with transcription. So in bacteria, this mRNA translation will start while transcription is still going. And truthfully, it's all kind of a continual process, but a single mRNA can be fed through more than one ribosome at the same time. That allows many protein molecules over here to be made, okay? And so you've got a bunch of these guys going on, and instead of working in the 2 directions, it's working in many directions here, basically, if you wanted to look at it that way, or you've got several of them attaching and going at the same time. It's almost like a little assembly line, I guess, for making proteins. Eukaryotes cannot do this because of the location. Bacteria don't have a nucleus, so transcription and translation can occur in the cytoplasm at the same time in the same place, okay? The processes are split because transcription for eukaryotes occurs in the nucleus and translation in the cytoplasm. But bacteria, they don't have to worry about that. It can occur all, and everybody can work at the same time because they're in the same place. So it's much more efficient. So you are making all of these proteins all at one time. This is a really good comparison contrast chart. I'm not going to read it all to you. I don't do that. But I always include them because I want to point them out and make sure you use these to study from. Use the pictures and the tables in your textbook. It is so helpful. Read them, try to understand what's going on in those processes, okay? So now the mRNA is going to leave the nucleus. We went through transcription where DNA was converted from the language of DNA into RNA in the nucleus. It's now the single-stranded mRNA. It's going to leave the nucleus. It's small enough to do so. It can go through the pores of the nuclear membrane, and it's going to go onto the cytoplasm. And the two mRNA subunits here are going to come together, or the two ribosomal subunits, sorry, are going to come together and form this little apparatus here. Okay, they're going to have attachment sites for your mRNA, and that's going to run through here like a ticker tape. It's going to start at the AUG and run through, and is going to be red. You're going to have your codons on those, and they're going to be matched up by the anticodons here on your tRNA. It's going to be read 5' to 3'. And the first one is going to be your start codon, AUG, which tells the tRNA to go and get methionine, or for bacteria, sometimes it's formal methionine. These little purple blocks represent your amino acids. So the tRNA is going to go, it's going to arrive in this site here after it picks it up and drop it off. It's going to have those anticodons. Remember, those are going to be complementary to the mRNA. You're going to have the P site. So here's the P site right here, and the A site, and that's going to guide the tRNA to enter. Then it's going to drop off the amino acids, which are going to form a peptide bond with the tRNA2's amino acid when it comes, and then they're going to leave via the E or exit site. All right, so the tRNA that's complementary to that first mRNA codon AUG, the anticodon is going to be the UAC. That's going to be the first to occupy this P site, as we just said. And then it's going to be where we have what we call the reading frame, which is what's reading the three-letter codons is going to shift, okay, to the right of the mRNA from one codon to the next. That's going to be your tRNA2, okay? And so that's going to leave that space. When the first one moves to the next site, it's going to leave the space for the next tRNA to come in. We're going to form this peptide bond here, as we said a moment ago, between the two. It's going to shift over and go to the exit site. And we're going to be forming a polypeptide as this continues. Here you can see the actual exit or discharge of the tRNA1 is leaving the exit site here. Notice that there is no amino acid now attached to that tRNA. It's going to stay here in order to make your polypeptide or your protein. tRNA2 then can slide over to this particular site. So now your tRNA2 is going to move over to the P site here. That's going to leave your A site open for tRNA number 3 to come. It's going to match its anticodon with this codon here, pick up the appropriate amino acid right here, which is proline, and it's going to come and it's going to bind there. And we're going to continue on with this process. That peptide bond now is going to be formed between the amino acids number 2 and 3. This particular tRNA2 is going to leave via the exit site. tRNA3 is going to shift over and leave the A site open for tRNA number 4. This picture is showing that continued process of elongation. We've got that tRNA4 is going to slide on over, and again, this is going to keep continuing until the stop codon is reached. Here you can see the illustration of that. There's that tRNA4 coming. It's coding for alanine, which is going to fill that A site. This will then join with this particular amino acid, form another peptide bond. And again, this will just repeat until we reach the stop codon, which is going to be different for every protein. Depends on the number of amino acids that you need to form the protein. When we reach that stop codon, you're going to have what we call termination. There are three codes here, three codons that mean stop: UAA, UAG, and UGA. Unlike the start codon, these do not actually code for an amino acid. AUG, remember, codes for methionine. So this amino acid number 1 is actually methionine, where the start codon occurs. But the stop codon is not going to actually get an amino acid. It's going to say, stop the process right here. We're done making our polypeptide. Again, this is one of those good summary slides. Remember that your start codon is AUG. It can be methionine, but in bacteria, that can be formal methionine. Eukaryotes mRNA is going to code for only one protein, but bacteria can code for several different proteins. Bacteria can do transcription and translation simultaneously because it occurs in the cytoplasm. Eukaryotes cannot because transcription occurs in the nucleus, translation in the cytoplasm. Bacteria have repeated triplet codons. Eukaryotes now have what we call introns that actually do not code for proteins, and these are removed by what we call the spliceosome. This is a little counterintuitive. Introns sounds like something that would stay, but the introns are actually what are taken out, and the exons are actually the portions that are going to be translated into the protein that stay in there. Okay, and then eukaryotes are also going to require some processing after translation, like adding a 5' cap or a poly A tail.